Today, I will be presenting on a recent study published in April of 2022 called Prion protein monoclonal antibody therapy for Creutzfeldt-Jakob disease, evaluation of a first-in-human treatment program. So first, what are prions? Prions are proteins that naturally occur in the plasma membrane in the central nervous system. Their entire purpose is not fully known, but some recent studies have attributed functions to prions such as neuron growth, synaptic plasticity, and even learning and memory and sleep patterns. However, these proteins are also unfortunately prone to misfolding, which then triggers the other normal prion proteins to misfold as well. This progresses and eventually causes brain damage. Abnormal prions can remain dormant in the brain for a long time, but once they start to act, the disease progresses very rapidly. These diseases are rare, but always fatal, and there are currently no effective therapies. Many cases of prion diseases are considered to be sporadic, as the exact cause is unclear. Others are infectious and are typically spread by the consumption of the meat from an animal that was infected, although they can also be spread through invasive medical procedures. Additionally, it is estimated that 10 to 15% of all cases are genetic. But specifically relating to this study, we will be discussing Creutzfeldt-Jakob disease, which some may know as mad cow disease. Although I do want to make a quick distinction that mad cow disease, when transferred to humans, causes variant Creutzfeldt-Jakob disease, which is very similar but slightly different than typical Creutzfeldt-Jakob disease. Typical Creutzfeldt-Jakob disease is considered to be sporadic. However, for this study, they had patients with both sporadic and variant Creutzfeldt-Jakob disease. So for the sake of this report, I will just be referring to it as Creutzfeldt-Jakob disease, or the abbreviation CJD. The research team, which included both medical doctors and other scientists, generated a fully humanized anti-prion monoclonal antibody, which they called PRN-100. For anyone who doesn't know, a monoclonal antibody is a molecule engineered in a laboratory to act as a replacement antibody to attach to unwanted cells in the body, or in this case, it was an anti-prion protein antibody, so its purpose was to attack those proteins in these patients' brains. So during the study, 6 patients were given intravenous PRN-100 every 2 weeks until death or withdrawal from the program. They closely monitored these patients for any side effects and also assessed the progression of their condition with the official prion disease motor, rating, and cognitive scale. They then compared these patients with untreated natural history controls. They also were able to perform autopsies in 2 patients and compare the findings. Their objective in this study was to hopefully slow or even halt symptoms associated with CJD. The results from this study showed there were no clinically significant side effects and the drug was well tolerated. Although the patients still displayed progressive neurological decline, the prion disease rating scale scores did appear to stabilize in 3 patients once the target dose was reached. Additionally, an autopsy of 1 patient's brain showed strong evidence of the target drug effect, with reduction in prion activity in the parietal cortex, the occipital cortex, and altered distribution in subventricular areas. The autopsy of the other patient also showed altered distribution of prion activity in subventricular areas, even though he only received 1 full target dose. In the figure on the screen, you can see prion labeling in the brain of a treated patient and in the brain of an untreated patient. In pictures A and B, around the cerebral aqueduct, and in C, around the lateral ventricle, you can see the less concentrated prions compared to the untreated patient in pictures E, F, and G. The prions are stained blue and are much more washed out in the treated patient. In pictures D for the treated patient and H for the untreated patient, you can see a difference in the parietal cortex. In the treated patient, it is much more washed out with a loss of the granular structure of prion proteins. I think the results from this study were incredibly promising, considering this was the first ever experimental treatment for a prion disease in humans. While the study was extremely limited in its scope by only testing PRN-100 in 6 patients at a mid to late stage of CJD, its results indicate a need for further testing of the drug. Not only would this drug fulfill the need for prion disease therapies, these studies could also help further knowledge in treating other neurodegenerative diseases such as Alzheimer's or other dementias.